Just a little background for those of you who don't know in the lab, even though it's kind of been announced here and there. We have two high school interns from High Tech High, Stella and Emily. This is their 5th week here, so this is their 2nd to last day. And in the past couple of weeks, they've been helping me and Swastik a lot with kind of our projects. And so this is kind of the culmination of everything they've been working on. And so we're super excited that you could all join us to see what they've accomplished and learned in the past 5 weeks. I also wanted to welcome 2 of their teachers who are here, right here, and then also Emily's family, and then also I think Stella's dad is online as well. So welcome to everyone to Scripps if you haven't been here already. Hopefully you guys enjoy their presentations. Without further ado, I'll let Emily take it away.
Hi everybody, my name is Emily Cho. I'm a junior at High Tech High International, and for the 5 weeks I've been here, I worked with my mentor Swastik for the expression and purification of SOSIP for HIV vaccine development. A little background about who I am. I'm from South Korea. I'm very in touch with my culture. I love it. It's my favorite place on earth. This is the Mr. Sunshine Land studio that I went to about 3 years ago in Nonsan. But I was raised and born in San Diego, my entire life. And I'm a junior at High Tech International, and this is my class at our Bay Area trip, it was 2026, and then this is a photo of me and Stella together exploring colleges. And then in my free time, I do a lot of figure skating. I've been figure skating for about 12 years. I've done a lot of competitions, tests, and this is me at my Christmas show. And then I also spend a lot of my time volunteering. I volunteer in the mother-baby unit for the Scripps Memorial Hospital over at La Jolla. And me and my friends, I have a very tight group of friends, and we run a club called Smart Cookies, where we fundraise for homeless children's education and tutor elementary schoolers after school at our local elementary school. And then I listen to a lot of different music types, but this is me at my favorite group's concert in 2022. And this is my brother. He's here today. He's my biggest inspiration as to why I'm studying the public health and the human body development about the brain and everything. So I would like to acknowledge him for that. And then this is a video of me falling, just for some. Jumping into what I did for the 5 weeks I've been here, I worked with HIV engineered trimer called SOSIP V9, 2 types called BG505 and GT1.1. I worked to verify the immunogenicity of SOSIP, and then did some MPEM with an antibody and antigen complex that I made throughout the weeks I've been here. These are the more specific steps that I took to do this project, and I purified and isolated the protein, and then made a complex with the antibodies, and then used some negative stain electron microscopy to visualize the protein complex. A little background as to what HIV is. It is the human immunodeficiency virus. There have been about 630,000 deaths as of 2023 because of HIV. And how it works, it weakens our immune system and leads further into AIDS because it targets cells such as the macrophages and T lymphocytes, which kill viral particles and bacterial cells to maintain our immune system. But HIV's surface glycoproteins called GP120 and GP41 bind to the CD4 copies that the macrophages and T lymphocytes have a lot of on their surfaces. GP120 binds to the CD4 copies, but then it also triggers GP41, which mediates the fusion between the viral cell and host cell to infect them, which is how they get infected. And then I worked with SOSIP and the broadly neutralizing antibodies. So SOSIP is the trimeric protein based on the HIV-1 envelope glycoprotein with stabilizing mutations to make sure that the it stays in the correct conformation. And then it's used to mimic the viral spike structure and elicit broadly neutralizing antibodies, and those recognize and block the spread of HIV-1. This is a little graphic about how broadly neutralizing antibodies prevent downstream conformational changes and viral fusion. first it's in the pre-triggered state, and then upon binding to CD4, there's a change in conformation, and then it binds to its co-receptor CCR4, CXCR5, I think, and then finally fuses into the cell and infects. So if the antibody sticks to the CD4 binding site in the site in the first state, it prevents all the other states from happening and infecting our cells. I used an analogy that my teacher gave me where I saw our host cell as a surface, and then the HIV cell as a piece of gum, and then body neutralizing antibody came in and got rid of the stickiness of the gum so it can't stick on the surface, but like water, something. That's an analogy I used to help understand it a bit better. Now jumping into the steps of my project, I first started with bacterial transformation. I had to transform BG505 and GT1 plasmids into bacterial cells, and then we inoculated 1 liter flasks of LB with one specific colony that we grew on the plates. Those are my results. And we transformed the bacterial cells instead of just throwing in the plasmid into the LB broth, because any colony that grows on the plasmid has the plasmid of interest because it grew on a plate with antibiotic resistance. And then with this, after overnight growth in an incubator, we used it for MaxiPreps. So the MaxiPreps was for me to produce DNA and then express protein. We MaxiPrepped to isolate the large quantities of the high purity DNA, and then we transfected them into HEC293F cells to express the protein. We used HEC293F cells because it stands for human embryonic kidney cells, and they're mammalian, so they would first of all express it in large quantities, and then they would because they're human cells, they have the proper glycans and sugars to keep it in the correct conformation. Oh yeah, and then we also transfected a mixture of the BG505 with furin and PEI. The furin cleaved the on-trimer, so it's in the correct conformation and folding along with the factors with the HEC293F cells. And then the PEI formed a complex for the DNA to pass through the cell membrane by coating the DNA with a charged shell. And then we had to purify the dissociative, and then we did that over PGT145 columns to isolate it. And we did this because PGT145 binds to dissociative, and we used an elution buffer we used TBS to get rid of all the other contaminants and the cell debris that's not the pure dissociative, and then we use an elution buffer to disrupt the interaction between PGT145 and dissociative to get just the dissociative out, the pure dissociative. And then we had to verify that the protein was properly purified and check its size through some gels. So we ran an SDS page and BM page analysis of each purification. And then these are the results. It's kind of blurry, but SDS is a detergent that denatures the protein and reduces them to break disulfide bonds. So it is useful in checking its purification because it differentiates based on the size and shows the monomers and confirms the subunits, which in our case is GP120, GP41. So it confirms the presence of those. And then the BN page is a blue native page, and it doesn't degrade protein, but it confirms that dissociative is properly assembling. And then this is a little graphic about how our cells produce antibodies before jumping to the next section. When it comes in contact with an antigen, it starts producing the plasma cell starts producing antibodies to the disrupting pathogen. So I started working more with Andy with this one. I was working with Jeff for the previous steps with the other labs, but we took blood samples from an immunization trial, and then we IgG isolated it, and then purified the Fabs, and we digested it, and then we complexed it with our antigen, which was GT1.1. We did great preparation. He put it under the electron microscopy, and then gave me the data processing and the analysis, and we looked over that together. This is some photos that I have of me of the process. This is me isolating the Fab and complexing it with the antigen, and then this is how big the copper grid was. I didn't expect it to be that small when Andy showed me like a photo of it, this. He showed me this first, but we used urinal formate to stain the copper grid, and then did the whole process with our protein, staining that too. And this is what the complex we made looks like under the microscope. These white little sections are all the Fab and antigen complexes, and then each particle was extracted from the micrograph and compared to others, and those with similar shapes and view were averaged together in the same class to improve the level of detail, and dissimilar particles were sorted into different classes, shown right here in the 2D class average. So you can see that there's the two antibodies here, and then an antigen. And then the 2D images are converted into 3D, and samples where different antibodies are mixed. Each can be sorted into a 3D space to create multiple 3D maps that represent the overall shape of the sample. And that was my final result. This 3D model was my final result. It showed that my vaccine trial was successful in generating antibodies for this specific antigen. I validated GT1.1 as an immunogen, and I made sure there were at least 3 antibody binding sites. There were more than 1 antibodies because this was taken from an actual blood sample from an immunization trial, so it produced multiple different antibodies, but it confirmed that the antigen had at least 3 binding sites for that. How was my work here impactful to the lab and just the work of public health? Through a negative stain, I was able to verify the structural integrity and visualize the antibody binding, and being able to see whether or not GT1.1 forms the correct trimeric structure. So we see whether or not it falls apart or stays intact, and which determines its candidacy for immunogenicity. And visualizing the antibody shows if there is proper binding at the key sites, such as the CD4 binding site, which validates that GT1.1 is displaying the proper targets for vaccine design. And then negative stain is quality controls before cryo-EM, electron microscopy, and immunization, because it verifies the structure before it's put under for a higher resolution visualization. And then all of that plays a role in supporting rational vaccine design. That's what I did for the 5 weeks I've been here. I would like to make some acknowledgements. I would like to acknowledge Andrew for making me feel so special in this lab and giving me an opportunity to be here. Jeff, because I worked with him for the first few weeks I was here. I asked a ton of questions. He made me feel safe asking questions, even though I kept pushing after getting an answer for one. Morgan helped me put the SciShow together. He let me practice even though I wasn't his intern, and gave me a lot of feedback. It was very helpful, and I felt very comfortable, a lot more comfortable than I would have felt because of the practice he let me. Stella, because I had a friend here and had a lot of fun while doing actual work and learning. Gabe, because he introduced me to the project. He made me help me understand and help me overall with the project. Swastik made sure I was learning the entire time, and I really appreciate that. I don't think I spent one day here where I wasn't learning something new, and I think I understand all of the material that I just presented very well. And Andy, he helped me through the last few steps, but he made sure I was understanding how to do it and kind of letting me be in a lab environment, actually using the equipment and stuff like that. And Hannah, I reached out to her in July of 2024, and she remembered that I reached out to her, and when internship season was coming around, she remembered and emailed me and to give me a spot here. And I would like to thank everyone else in the lab for making me feel so welcome and working here. Thank you very much.